Welcome everyone again to another episode of the ideaXme show that profiles the human beings behind the really big ideas that are shaping our world, inspiring future creators, and for all those that like really great stories. I'm Ira Pastor. I'm your health, aging, and longevity ambassador. Today we are going to step in a direction that we have not gone in yet and talk about our largest organ, the skin. While many people might sort of look at the skin as just sort of another layer of tissue, at the end of the day, it has to be remembered that in addition to being the body's largest organ, it represents this really complex, biological membrane that contains billions of cells, miles of blood vessels and nerves, billions of pores, and ultimately is intricately connected to everything going on below. And constantly interacting and communicating back and forth between the other 30 trillion or so cells that make up your body, either in protecting us from external threats and endogenous stimuli, as well as, everything that percolates upwards, whether that's inflammatory factors, oxidation byproducts, epigenetic factors, and so forth, that affect its appearance, biology, health, and so forth. So today, to go further into this area and into some very exciting work in the area of regenerative medicine, I'm happy to say that we're joined by Dr. Luis Garza, Associate Professor, Department of Dermatology at Johns Hopkins School of Medicine. Dr. Garza received his undergraduate degree in neurobiology from Cornell and his joint MD PhD from here in Philadelphia at University of Pennsylvania School of Medicine. Dr. Garza completed his residency in dermatology at University of Michigan. He performed his fellowship in dermatology at UPenn, and he was involved in discovering at that time really the intricate connections between prostaglandin synthesis and male pattern baldness. He joined Hopkins in 2009, and his areas of clinical expertise right now include wound care and general dermatology. And in addition to treating patients, he runs a molecular biology laboratory at Johns Hopkins that studies skin stem cells and wound healing with an emphasis on identifying sort of a next generation of wound healing therapeutics and diagnostics. The current focus of his lab is on understanding what controls and maintains skin identity, as well as how, sort of connected to this theme, during wounding, skin identity is typically lost via scarring, ultimately in certain situations can yield complete regeneration. Understanding these questions will have broad significance to regeneration and stem cell biology in multiple organs, and understanding wound healing processes that can reinitiate embryonic developmental patterns and may eventually lead to insights to trigger, ultimately how to trigger regrowth of much more complex tissues, organs, limbs, and so forth. Dr. Garza is Associate Editor for the Journal of Investigative Dermatology. He's a member of the American Academy of Dermatology, the Society for Investigative Dermatology, and the North American Clinical Dermatology Society. All that being said, Dr. Garza, thanks for taking the time to come on the show today.
It's my pleasure. Thank you for the invitation.
Absolutely. Absolutely. So, we typically start off by giving our guests the floor just to learn a little bit more about you, sort of, your background, how you got interested in health, medicine, dermatology, and ultimately sort of how you've segued now into this fascinating area of complex regenerative biology.
Sure. Yeah. So basically, I've always been intrigued with research as a way to kind of uncover new knowledge. To me, kind of the beautiful thing is that if you discover something that's true here in Baltimore or in Philadelphia, somebody else can do the exact same experiment and they'll discover it's true in like Australia or China. So we're trying to find these kind of deeper truths really that are going to be correct like anywhere in the universe, which is really fun. A fun task about science. So I think fundamentally that's what I'm intellectually excited about. And then what better truths to learn than those that relate to human health? And so that's been a focus of mine for a while. And lately I've been really fascinated with regenerative medicine because really the miracle of developmental biology. So, you started off as a single cell in your mother's womb, and now you're running a podcast that has listeners, I'm sure, all over the world. So that's an amazing kind of feat of biology that can happen, that you can go from one cell to the complex ideas of Einstein or something like that, and that happens through developmental biology. And to me, the question is, can we harness developmental biology as a therapy in medicine? Because there's a lot of human disease, and medicines help for sure, but none of them are as powerful as our own bodies. And the question is, can we harness the power of our body as therapy?
looking at sort of the focus of your lab, you're involved in really, two fascinating areas. On one hand, wound healing. On the other hand, regenerative biology. And, in many ways, these are a double-edged sword when it comes to humans, because, I point out on some other shows that, we have, we're a species that, we bleed very rapidly. We die very rapidly from loss of blood. And, evolution and the powers that be have granted us this, pretty decent wound healing response, which, doesn't always make for the best regeneration. There's other species, and you talk about them a bit in your research, that they're very good at it. Instead of laying down the scar tissue, are very good at doing more complete regeneration. Can you talk a little bit how you balance these two parts of your research program, because they are, while both involved in sort of regeneration processes, they yield different things, and how we as humans can sort of, in your research, balance them out.
Yeah, I think there's two kind of dichotomies you're talking about. One is, in wound healing, and the other are the focuses we have in the lab. So in wound healing, I think the question is, how quickly do you want to patch the hole, like you say, and how much do you want to regenerate the structures that used to be there? And you're right, those are kind of somewhat competing aims. And it's a very interesting question about how evolutionarily we've gone about addressing that. Because you can imagine that if you lose too much function, even though you repair, you're at a disadvantage. The other kind of related trade-offs people believe is that there could be a trade-off between cancer and regeneration, because there's a lot of pathways that are involved in regeneration that are also kind of hyperactive in cancer. Because in a sense, a cancer is kind of like organogenesis gone, severely awry, where you kind of turn back the developmental clock to the detriment of a person. So people kind of wonder whether it's kind of a yin and yang in nature, and that humans might have less of a regenerative ability to try to suppress cancer. But that's kind of speculation. Nobody knows 100% for sure. For that model, what we're looking at is wound healing. And in that context, we're asking if we do these enormous wounds in the back of a mouse, and mostly the mouse heals by patching the hole, but in the very center of that wound, the mice make a new hair follicle from nothing. So the cells kind of crawl across, and they act as if they were in their mother's womb again, and they make a new hair follicle. And we find, although it's small, that example of organogenesis is really intriguing to us, and that's a big focus in the lab. Interestingly for that, we noticed that there's not huge trade-offs between wound closure and regeneration. At first we thought that, yeah, there would be, if you patch the hole much quicker, you would have major differences in the amount of regeneration you have.
Okay.
We're finding a lot of examples where the regeneration is different, but the time you patch the hole is similar. So it's not bearing out as much as we thought to be the case, but it certainly, theoretically, it continues to be possible. So anyway, half the lab studies that. So then the second dichotomy is what we study. The other half of the lab looks to say, all right, let's look at regeneration in a different way. So, and I'll explain that in a second. So in the first part of the lab, part of the reason we're excited about it is it turns out that hair follicle development and limb development overlap a lot. So that if, for example, if you have mice with a couple of mutations in important genes, like one gene's called P63, another gene's called beta-catenin, those mice don't develop hair follicles, and they don't develop limbs. Because the process of developing a new hair follicle and developing a new limb are very similar. And so what we think is that if we can understand how to make a new hair follicle in the middle of the wound, we might get closer to forming a new limb, like you were referring to earlier. So that's that half of the lab. The other half of the lab, to kind of try to tie them together, studies ask this question: okay, well, if we can't regrow a whole limb now, can we at least regrow this thick type of skin on our palms and soles? So that an amputee who's wearing a prosthetic will have skin that's pressure adaptive and friction adaptive at the stump site. So, most of your listeners didn't think twice when they put on their shoes this morning, because their skin is adapted to bear weight at the soles. And so the question is, can we enhance those properties at the stump site of an amputee? So I think the dichotomy from my lab there are we're really focused on really limb regeneration in a way, and in
Mm-hmm.
both parts of the lab. And in one context, we're looking in wound healing and asking the hair follicle to be a proxy to like try to learn more about appendage regeneration. And then in the case of the second part of the lab, we're saying, can we help even humans right now? We're doing clinical trials that we can talk about later,
Sure.
using stem cells to try to change the skin at the stump site. So those are the two things we focus on.
And continuing along that theme, you had this really fascinating paper a couple months ago. It was in the press. You talked about self non-coding double-stranded RNAs. And we can go into a little bit of what that is, but talking about sort of the biologic factors that are involved in some of this regeneration per the hair follicle. And then also, you discussed a bit, not just sort of the biological, sort of the biochemical side of that research, but also you mentioned how, there are, the hypothesis there may be in some of these sort of skincare type interventions that are not biochemical, but maybe biophysical in nature, whether they're lasers or dermal rollers or whatever, that there may also be sort of a biophysical side to this regeneration. Can you talk a little bit about the paper and sort of what you're discovering?
Yeah, so we were really excited about that paper. It took many years of work, and it capitalized on even more years of work. We've been kind of working on this general problem for about 10 years now, here at Hopkins. So and that was pertaining to that first part of the lab that studies this wound healing event in mice where they regrow the new hair follicles, and we ended up being able to tie that to some regeneration we see in people. So I'm happy to explain it.
Sure.
So in mice, I described that we do these large wounds in the back of the mice, and at the very center, the cells crawl across and they start to almost kind of like the Terminator. You know how like in the movie, the Terminator is like a puddle of like silver looking stuff, and he can self-assemble into like the Terminator no matter kind of what you do to them.
Terminator 2.
Yeah, and that's really exciting about like stem cells is they kind of have a similar property. Like stem cells can be like a cloudy solution. I mean, you squirt them into the right place and they can self-organize into an organ. And that's the process that's really happening in this wound-induced hair neogenesis. So these cells are like single cells, they're crawling across a wound, and they act together in this beautiful dance to create this new hair follicle that ends up being an anchor for blood vessels and nerves, like you said at the beginning of the intro, to kind of be a complex tissue. So we've been really curious about that, and we started off the lab, many years ago, trying to compare mice that had really good regeneration and mice that had poor regeneration. So the ones that made, the same type of wound, there's some mice that are inbred that are poor regenerators, and they'll make very few hair follicles at the end. And there's other more outbred strains that aren't as inbred, and they're more healthy, and they, after the same wound, they can make a lot of new hair follicles. And so we compared those two mice before they started the regeneration. So right as the wound was closing, but before any of this organogenesis had happened, to ask what's the signature of a wound that can make really good regeneration. And when we did that, we found there was a really strong signature for this gene called toll receptor 3. And I'll explain what that does. It's a receptor for double-stranded RNA. So it used to be thought that your body needed receptors like toll-3 to as part of what's called your innate immunity. So you have kind of two types of immune systems. You have like the adaptive immune system that we all know really well, which is like your T cells and B cells, and these are ways. These are why you get vaccinated, to try to turn on these pathways to protect you from infections. But then you also have this very ancient immune system that even like flies have, that's called your innate immunity. And for those, there's receptors that recognize a whole broad range of potential pathogens or infectious agents. And toll receptor 3 was supposed to be and it really is a gene that recognizes a lot of viruses. In particular, it's thought to recognize viruses that have 2 strands of RNA. So normally in your body, you have 1 strand of RNA that makes proteins they're building blocks to your body. This toll receptor 3 is thought to recognize double-strand RNA, which has 2 strands. And it was previously thought to be limited to viruses. But basically what we found is, and others have I found this also for sure, is that double-strand RNA, it's not only kind of a marker for infection, it's also a marker for damage. Like when a cell gets busted up, like the RNA might get totally misfolded and some of it becomes double-strand That's the current hypothesis in the field, like canonical view, is that
Mm-hmm.
it's released in this kind of tumult of damage, and where you have this double-stranded RNA, and then that gets sensed, and we showed that that double-stranded RNA ends up being a stimulus for regeneration. So if we even just take two mice, one we give it just a vehicle, and the second we actually intentionally scored on double-stranded RNA, we can dramatically increase the amount of regeneration that mouse has, the number of new hair follicles it forms. And so we've been really curious about that pathway. We've done a lot of work on it. And the paper that just came out was us looking at another facet of that, saying what happens downstream of the double-stranded RNA.
Okay.
And also, we were curious, is this pathway important also in humans? So to look at humans, what we did is we collaborated with groups here at Hopkins that are clinical dermatologists that are doing these treatments every day, such as laser facial rejuvenation.
Okay.
So like if people have fine wrinkles on their face and they want to get rid of them, one of the therapies they can have is getting squirted with these lasers that stimulate a regeneration response in a way we're not totally sure of. So they have mechanisms that haven't been completely elucidated. And so to help discover that, we collaborated with this large group at Hopkins. We're really grateful for their help. And they, together, we basically took a baseline biopsy of people. So we knew what their photo damaged skin was like beforehand. And then we gave them this laser rejuvenation therapy and did a second
Mm-hmm.
biopsy afterwards. And then with those biopsies, we queried every single gene's gene expression, like what's happening in that by doing a microarray, it's called. So
Mm-hmm.
we were kind of analyzing all the potential genes that got turned on and off. And we plot them on a computer to say, what are the ones that have the highest significance and what are the ones that have the highest fold change. And so what the crazy thing we found when we were doing this human work is it was the exact same pathway we saw in the mice, that this double-stranded RNA pathway is dramatically induced by lasers. And so that first was really exciting because it showed that this isn't just a laboratory phenomenon. This is happening every day when people are getting their laser treatment, is they're turning on this double-stranded RNA pathway. And then we went on in the paper, and I can describe later how we discovered what was downstream of that and how that's actually also important for human health.
You spurred something in mind before you mentioned the stem cells and the Terminator bit, and I guess this goes back to your theme of identity and patterning, because obviously we've spoken in the past on the show about we have we made these 30 trillion cells, as you point out earlier on. It's all the same genome, and little differences in gene expression here and there dictate if something becomes a cardiomyocyte or a neuron or whatever. But then, as one goes along in the human body, this heterogeneity continues. And, obviously the skin on top of my arm here, which has hair on it, is different than the skin on my palm, which never grows hair, and so forth. I was just wondering, because this whole theme of sort of identity and position of where stuff goes is always a big theme. And, once again, think about the Terminator of having an arm reform as opposed to a leg or whatever. Are you working a lot with sort of these positional identity genes, some of the WNTs and some of these other components that are involved in sort of dictating what happens at each step of the process as that regeneration occurs?
Yeah, definitely. Yeah. So then that's a really fun question you bring up. Yeah, the family of genes we look at that's the most directly related to that are these genes called the homeobox genes.
Yeah.
So in like Drosophila, for example, there's really, really amazing things that happen when you screw up these homeobox genes. So for example, one of the best known ones is called antennapedia. And so if you mutate antennapedia, instead of having an antenna, the Drosophila or the fly will have an arm coming out of its head. Or like trithorax will have it'll have not just two wings, it'll have like extra 4 pair of wings. So there's like a lot of really amazing homeoboxes, and it turns out that a colleague in my field, Howard Chang, who worked at Stanford, was one of the first people to show if you take biopsies from different people's skin, like let's say going from their tip of their fingers up to their arm, up to their chest, and you take out the fibroblasts, so the part of the skin that's your dermis, that's kind of like the part that makes leather basically from a cow.
Mm-hmm.
If you take those cells out and you put them in culture, you can passage them from even 10 you can keep them in tissue culture for many months. And if you query them, they still have super strong expression of their homeobox. So your body actually, the cells remember exactly where they came from. There's a GPS where your cells know their positional location. And the even cooler thing is on your DNA, the position on the DNA strand actually even matches the position, the physical position on your body of those cells where they express it. They call that collinearity. So your DNA is this kind of chemical structure that's super huge and long. It turns out where the homeobox is on that DNA also reflects where it falls along your arm, which is totally wild. Those two physicalities could have that symmetry. So yeah, positional identity is super cool. We're really interested in it. We're using that to try to help the amputees. What we're trying to now basically tell them, look, by using cell therapy, we want to convince the skin that the stump site is actually positionally the palm and the sole, so it can have those properties of friction resistance and pressure adaptation. So yeah, we think about positional identity a lot. It's certainly really important.
Yeah, absolutely. It's a fascinating area, and I'm glad to see that it's part of sort of the continuum that you're working on, because it's I know that people have been studying sort of the concept for 100 years, how that all happened so perfectly, but it's nice to see sort of it being weaved into the modalities and the thinking that you're working on. So speaking of this, here we are in 2019. Obviously, you're doing so many things. Take us out a little bit. What are you hoping clinically? Where are you hoping to go? Is it more skincare, or are you focusing more heavily on the more complex regeneration? If we come back in 20 years, what do you hope to? What types of therapies do you foresee coming to fruition from your work?
So basically, we're definitely excited about continuing, what we're looking at double-stranded RNA. Kind of, there's a lot of important basic questions there that can also lead to therapy. So it could be like basically from that laser study, we found out that double-stranded RNA turns on retinoic acid synthesis, which is another important chemical in your body that's actually also important for patterning. And retinoic, in addition to being important for patterning, retinoic acid also is a therapy. So it turns out a lot of people who go to the drugstore buy retinol or retinols, like retinol skin creams, and those creams are supposed to enhance regeneration of the skin.
Yeah.
And we actually found that double-stranded RNA turns on your local production of that retinoic acid and retinols.
Okay.
So basically, we think that could be a product, is working with industry to try to find the optimum formulation of double-stranded RNA that can help people with photorejuvenation. So that's a small kind of thing, but might make an important difference in people's lives that we're interested in pursuing. And then on the stump project, we're doing clinical trials right now. We're actually injecting these cells into amputees to try to help them. We're measuring to see if their skin gets thicker, if they can use their prosthetics more frequently, using like activity monitors, doing quality of life questionnaires. We're working with the Department of Defense to help wounded warriors that way. So we're going to be recruiting patients at Walter Reed, also in DC, also Naval Medical in San Diego, and Brooke Army in San Antonio. So we're really excited to continue collaborating with our Department of Defense colleagues and funders to try to make cellular therapy a reality for the wounded warrior and then amputee civilians. And then we think that cellular therapy could maybe be the beginning of a whole bunch of different applications. Because cells, they're probably going to be the therapy of tomorrow. Like right now, people take a pill whether you need it or not. Like, the pill doesn't know if you need it. It just does it because you stick it in your mouth, and you do that because your doctor tells you. And your doctor tells you because that's what they think that you need, but they don't know moment to moment if you need that medication.
Right.
The exciting thing about cells is they can sense what's happening around them, and then they can give the response that's required. So we think cells are going to be the therapy of tomorrow, and we're really excited to start that work.
we typically have a wrap-up question on the show where we sort of get back to something more personal, but it's really a question about influencers in your career to date. Obviously, you've probably met a lot of fascinating people along the way, and research and science and what have you. Any specific people that you might want to give a shout out to in terms of people that have kept you on this path? Obviously, biomedicine is tough. If there's a lot of failure in it, is there any specific people that guided you and said, if it wasn't for them, you would have gone into law or something totally different that you might want to shout out to?
Yeah, like I probably want to definitely give a lot of shout outs to my colleagues here at Hopkins, because they've been really helpful, really collaborative, and without them we couldn't have done none of this work. So my chairman, Siwan Kong is his name. He helped us even initiate the stump study that I told you about, and he's given us a lot of support. Our clinical collaborators, we couldn't have done this work without them. Like this Dr. Anna Chen kind of runs our translational unit, and then she helped with this laser study. Also, Mary Xu helped run it with another colleague named Nori Kim. There have been fantastic researchers in the lab that have helped, like Sashank Reddy was an MD PhD, is now plastic surgery faculty here, who helped out a lot. Amanda Nelson really pioneered a lot of our double-stranded work. The retinoic acid work was done by a guy named Dongwon Kim. We've had, it takes a village literally to do this stuff. So the first people I want to thank are all of our collaborators and like lab folks and clinicians. And then, general scientists that have been inspiring. I've had a lot of help definitely along the way from a lot of people that have been, really, really inspiring. Like beyond the ones I mentioned, like my former chairman at Michigan, John Voorhees, was super supportive. Then my chairman when I was at Penn, John Stanley, was also extremely supportive. So I'm really grateful for those guys. And they gave me a lot of really useful advice. And then there's of course scientists, awesome thing like Louis Pasteur. It's like there's this great book you might have heard about by Stokes called Pasteur's Triangle, where he says, you can have research that you can divide research into 4 quadrants. You can say, what research does it have a fundamental kind of basic importance to science? And yes or no. And then you can have another one that says, is there applied importance of this research? And so an example of like pure basic science would be like Niels Bohr, who like invented who discovered a lot of important things about the atomic structure. For purely applied research, you can think of like Thomas Edison. But then in that really sweet spot of yes to both was like Louis Pasteur, who discovered, super important things about fundamental immunology, but also invented vaccines, like the best medicine we have. So also I've had great inspirations from like the really incredible scientists that shaped what we all do today.
Wonderful. Wonderful. I appreciate it. You went into the other question I was going to ask you, but that was perfect. I usually put our guest in a time machine and I have them go meet somebody. So Louis Pasteur, would love to meet him too.
Yeah.
Dr. Garza, it's really been a pleasure having you today. Obviously, you have so much going on. Really appreciate your time and everything you do, as we say on the show, moving the human story forward and really changing the future. For everybody that is going to be listening to the show on the podcast or watching the episode, we're with Dr. Luis Garza, Associate Professor, Department of Dermatology at Johns Hopkins School of Medicine, wound healing, limb regeneration, really big picture things that are really going to be crafting the future of healthcare moving forward for degenerative diseases. Dr. Garza, thank you for joining us and thank you for everything you do.
Hey, it's my pleasure. Yeah, thanks for pushing science. Public communication and public advocacy is so important these days. Our public has to kind of really value what we do for us to continue, and we depend on their support and folks like you to communicate. So I'm very grateful for this opportunity, and also I want to congratulate you on your important work. Thank you.